Hello everyone. In this class, we are going to discuss about the overview of a protein purification. So here our aim is to isolate the protein of our interest from the biological samples. Biological samples they will be having around 10,000 to 20,000 different type of proteins, out of which we need to isolate the protein of our interest. So here in the protein purification, it is going to involve around 3 to 4 different type of the fractionation steps. And here in this method, 100% protein purification or the isolation is a difficult task. But fortunately, we can do any studies on proteins with the 5 to 10% contamination with the other proteins. For example, if you are using the proteins for the sequencing purpose means, so here the 90 percent purity is sufficient. But if you are going to use the proteins for the antibody rising in the mice or the rodent means, the 50% purity is also sufficient. But at the same time, if you are going to use the proteins for the enzymatic activity means, so here in the studies of the enzymatic activity, relatively impure sample is also sufficient. But if you are going to use the proteins for the any therapeutic use means, so here high percentage of the protein purity is required. So here therefore the degree of purity it is required for the purpose for which the proteins are going to be used. So here these are the contents which includes the introduction, cell disruption, and the production of initial crude extract where we are going to lyse the cells. After which the separation of a protein of our interest from the bulk proteins with the help of the salting out or the dialysis, and here which is followed by the high resolution fractionation methods which includes ion exchange chromatography, size exclusion chromatography, affinity chromatography, and at the end we are going to discuss about the separation of proteins on the page. So here, so here the proteins are important biomolecules made up of the series of amino acids which are linked one another with the help of the peptide bond. The protein purification is a series of processes intended to isolate one or few proteins from a complex mixtures, usually cells, tissues, or the whole organisms. Why it is important? It is important for the characterization of structure, function, and interaction studies of a protein of our interest. And here at the same time, the protein purification it might be preparative or analytical. Preparative means, so here at the mass level we are going to isolate the proteins, but the analytical means, so here we are going to use it for the research purposes. So here the first step in the protein purification is the cell destruction and production of the initial products tag. So here the biological sample. So here the first step is we need to lyse the cells with the help of the suitable buffer. So here for which we need to understand the composition of the extraction buffer. So here which includes, so here in the most of the cases we are going to use the TIS or the phosphate buffers of ionic strength 0.1 to 0.2 molar and the pH of around 7 to 8. And here in addition it is having the several other components which includes the antioxidants. So here we are going to use the dithiazole or the beta mercaptan ethanol. So it is because once the cells are going to be lysed, so here inside the cells usually the proteins they'll be exposed to the reducing environment. But here after the cell lysis they'll be immediately exposed to the oxidized environment. So here due to which the sulfidal groups in the proteins are going to be oxidized. So here to avoid that one we need to use the reducing agents such as dithiazole and the beta mercaptan. So here in addition we can also able to use the protease inhibitors because so here after the cell lysis whatever the proteolytic enzymes are there in the lysozymes they'll be released out, and here they'll be started degrading the proteins. So here to avoid this one the protein purification it is always done at the 4 degrees Celsius. At the same time we can also use a certain protease inhibitors. For example, diisopropyl phosphofluoridate, which is a serine protease inhibitor. One more example is pepstatin, which is a aspartic protease inhibitor. So here by adding these protease inhibitor, we can able to minimize the action of the proteolytic enzymes. And here in addition, we can also use the enzyme substrate and the cofactors. Enzyme substrates are going to bind to the enzyme's active site and going to stabilize the enzymes. At the same time, cofactor also gives stability to the enzymes, and also it is also helpful in screening the column fractions in the enzymatic activity also. And here in addition we can also use the EDTA, and here the EDTA, it is going to remove the divalent metal ions from the samples. So here, because these divalent metal ions are going to react with the sulfatyl groups in the proteins and going to form the mercaptides. To avoid this one, we are going to use the EDTA, which is going to remove the divalent metal ions. And here in the isolation of the proteins from the plant extract, there we are going to use the PVP, which is nothing but the polyvinyl pyrrolidine. This polyvinyl pyrrolidine, which is helpful in removing the phenolic compounds from the plant tissues, because the phenolic compounds, which includes p-hydroxybenzoic acid, are the tannins that bind to the proteins and going to result in the aggregation of the proteins. And here at the same time, these phenolic compounds, after the cell lysis, they will be oxidized by the enzyme called as the phenolic oxidase and going to form the quinones. These quinones are going to readily react with the proteins and going to form the cross-linking of the proteins and ultimately results in the protein aggregation. So here, for this reason, so here we need to remove the phenolic compounds. So here to do that one, we are going to add the polyvinyl pyrrolidine in the extraction of the proteins in the plant tissues. So here this PVP, it is going to absorb these phenolic compounds. So here after the absorption, these phenolic compounds are going to be removed by the centrifugation. So here these are the composition of the extraction buffer. So here now we need to discuss regarding the different type of the cell disruption methods. So here we can able to disrupt the cells either by using the blenders or by using the mortar and pestle along with the abrasives, or with the help of the French press, or with the help of the enzymatic methods, or with the help of the sonication. So here in the blenders, the domestic kitchen blenders also it can be used in the cell lysis of the mammalian tissues or the plant tissues also. So here this blender also it can be used in the mammalian tissues or the plant tissues, which is going to lyse the cells, and here after which the cell debris or the tissue debris it can be removed with the help of this centrifugation. And here in the this mortar and pestle also used in for the fungal samples or the plant samples also, for the lysis of the fungal samples or the plant samples also, along with the suitable abrasives such as the sand or the alumina. And here we can also able to lyse the cells with the help of the French press. And here this French press it is suitable for the microbial cells or these cell suspensions. So here in this case, as you can see in this image, so here the cell suspension are placed over here, and here it is passed through the small orifice in a high pressure. So here for this cell suspension, so here high pressure is applied, and here it is forced to move through this small orifice. So here once it is going to move through this orifice means, so here it is going to result in the cell rupture or the cell lysis. So here by providing the high pressure, so here the cells are going to be passed through the small orifice. So here once it is going to passes, the cells are going to lysis over here. So here this is in case of the French press, and it is suitable for the microbial cells or the some of the cell suspensions. So here the next method is with the help of the enzymatic method. So here the most of the cells, for example bacterial cells, yeast cells, or the fungal cells, they'll be having the cell wall along with the cell membrane. So here to remove this cell wall, we may need to use some of the enzymes. For example, for the bacterial cells. So in the bacterial cells, the gram-positive bacteria is there and the gram-negative bacteria is there. And here both of these bacteria it can be lysed with the help of the enzyme called as the lysozyme. This lysozyme which is going to cleave the peptidoglycan layer in the cell wall, and by doing so we can able to remove the cell wall, and especially in the gram positive bacteria. The gram positive bacteria it will be having a very thick peptidoglycan layer, and it can be cleaved with the help of the enzyme lysozyme. And here after which, after the separation of the cell wall, the bacterial cells now it will be having only the cell membrane, and here these cells further it can be ruptured with the help of the osmotic shock. But in case of the gram negative bacteria, In the gram-negative bacteria, the cell wall composition will be different, and here it will be hang the thin peptidoglycan layer, along with that it will be hang the lipopolysaccharide layer. And in this case also we can use the lysozyme along with the EDTA. EDTA, it is going to chelate the calcium ions, and here these calcium ions are removing calcium ions. It is going to destabilize the lipopolysaccharide layer, and here now the lysozyme, it is going to cleave the thin peptidoglycan layer. By doing so, we can also able to cleave the gram-negative bacteria. And here we can also cleave the cell wall of the yeast cells by using the zymolase. This zymolase, it will be having the beta 1,3 gluconase activity. And here the zymolase, it is used for the cleaving the cell wall of the yeast cells. Whereas the chitinase, it is going to be used for the removal of the cell wall from the fungal cells. So here these enzymes are commonly used, and at the same time one of the drawback is, so here these enzymes are they will be having the high price, and here they will be having the limited availability. At the same time, the addition of these enzymes, it is going to complicate the downstream processing, example the purification. But here these drawbacks, it can be minimized with by the immobilization of these enzymes. So here this is the enzymatic method. By using these enzymes, we can able to clear the cell wall. So here the cell wall will be removed, and here now the cells will be hanged only the cell membrane, and here further these cells, it can be ruptured easily with the help of the any osmotic shocks. So here it is in case of the enzymatic methods. And at the same time, we can also use the one more method called as the sonication. In this method, so here it is suitable for the suspension of the cells or the microbial cells, and here the cells are exposed to the ultrasounds of around 20 kilohertz for 30 to 60 seconds. And here in this case also the cells are going to be disrupted by the shear force, and here it is going to create the cavitation. With the help of the cavitation, the cells are going to be lysed over here. But in this case, in the sonication, where the ultrasounds are going to be used, and here it is going to result in the heat generation. And here, to avoid the heat generation, in this method it is done at the in the ice at the 4 degrees Celsius. At the same time, the small volume of the biological sample is sufficient for this process. Thank you.